If you fully understand the goal of meiosis and what is trying to be achieved, which is to make a haploid cell starting from a diploid cell, and to make sure that that cell has one copy of every chromosome, either the chromosome that came from the mother or the chromosome that came from the father of the organism making the gamete, then you can very easily draw a gamete starting with the initial cell. You shouldn't have to go through all of the stages of meiosis to figure out what is happening. So we need to follow a few rules if we're going to be able to do this. The first one is that there should only be one of each pair in the gamete. Now remember that we're going to start off with a diploid cell. So here, this is a diploid cell, it's 2N. There are 2 copies of every chromosome. So let's say this yellow one is chromosome number 1, the pink one is chromosome number 2, and the blue one is chromosome number 3. So we need to end up with an orange or yellow, a pink, and then a blue in the final gamete. And the other rule is that there should be no duplicated chromosomes. Now remember that a duplicated chromosome is that X shape, so no X shapes, because this is 2 sister chromatids. Now let's just take a pair of chromosomes through meiosis. So here's your starting cell. I'll draw one down here. We'll just draw 2 chromosomes, 1 homologous pair. That cell is going to go through S phase of interphase, and each one of those chromosomes will be duplicated. So we'll get the 2 X shapes. Now this is a homologous pair. Remember that homologous pairs are separated in meiosis. So in meiosis 1, this is when this happens, meiosis 1, the pairs are going to be separated. So this one's going to be separated from here. So the 2 resulting cells are like this. Then in meiosis 2, the sister chromatids are separated. So we end up with just one of the chromatids in each cell. And now there's crossing over happening in all of this, but in terms of what we should end up in the end compared with the start, it's just one of each pair. So down here, one of each homologous pair goes in each gamete, and there should be no duplicated chromosomes. Meiosis 1 figures out separating the homologous chromosomes, and meiosis 2 separates those sister chromatids. So if I'm to take this cell, for example, and this is the cell before it undergoes S phase of interphase, there's no X shapes. It's actually kind of easy to do. All I want to do is make sure I take one of those pairs. So there's going to be one of the yellow one, there's going to be one of the pink one, and there's going to be one of the blue one. Now this one, this is exactly the same cell. This is just after S phase, so it's the same cell. So the gamete, that's exactly the same. Why is that? Well, let's take it through meiosis 1 first here, which separates the homologous chromosomes. So let's just draw one of the cells that would have been made. The cell's going to get one of these X's like this, one pink one, and then it's going to get one blue one. This is meiosis 1 here. Meiosis 2 is going to separate those chromatids. So this is the gamete that is going to result from either of these cells. Let's follow the rules. We've only got one per pair. We've got one per pair here, one per pair, and we've got no X shapes, so no X shapes here. So very easy to do this if you understand what is happening. So let's do one practice. Okay, so here is a cell, and you need to ask yourself first, is this before or after S phase? So you're seeing the X shape, so this is after S phase. So this is before, right before we're going to go into meiosis. And how many homologous pairs are there here? Well, here's one homologous pair. Here's another homologous pair, 2 copies of chromosome number 2, and here's another homologous pair here, 3. So following the rules, there should be just one from each pair. Then there should be no X shapes. What are the possible gametes? See if you can draw all of the possible gametes. If not, just try and draw one gamete that's been made from this cell. Pause the video and see if you can do it. Well, I actually asked you to do something quite difficult here, but let's just draw one gamete first. So let's take one of these pairs and stick it in the gamete. Let's say it's this one here. Let's do this blue one here. So it's going to get one of the chromatids from this pair here. Remember, one of the pair, no X's. And then let's say it gets this orange one here. So it's going to get this one here. And then let's say it gets this light orange one there. So that's one possible gamete. The other possible gametes could be the other chromatids like this. Now there's often crossing over. Just a little recap on that. The chromosome pairs are going to line up next to each other, and the inside chromatids are going to swap a bit of genetic information. And so we end up with 4 unique sister chromatids instead of just 2 versions. And I haven't shown crossing over here, but if there was crossing over, we would have a bit of this reddish one in here, and we'd have a little bit of this green one in here. But I'm going to keep it simple right now. So one gamete is going to get this chromatid, this chromatid, this chromatid. The other gamete is going to get this chromatid, and this chromatid. And then we're just going to do the same again. So one's going to get this chromatid, the other's going to get that chromatid. And then of this one here, one's going to get the one on the right, and one's going to get the one on the left. And then this one down here, remember, one's going to get the one on the right, and the one's going to get the one on the left. And now this is without crossing over. But the important thing is, let's see if we've followed our rules. There is only one chromosome per pair. So only one member of the pair. So it's either going to be a green chromosome or a blue chromosome here. Okay, yeah, we've only got a blue chromosome. We haven't got a green one. And we actually also have a green or a blue one. We've got to make sure that we've got at least one of them. And the second rule is no X shapes, because in meiosis 2, we're going to get our chromosomes and we're going to pull the chromatids apart, just like we did in mitosis. So yeah, no X shapes either. Now this can be difficult to do depending on what cell you've started with, and just keep in mind that before S phase, that chromosome was just a single line like that. So you've got to ask yourself first, is the cell before or after S phase? If it's before S phase, duplicate the chromosomes. If it's after S phase, you're good to go.